we're working on at the end of class on Monday. Again, the problem with this one, or the prompt for this one, is you've got an ocean or a microorganism that's found on the hull of an ocean liner. How might, just based on that description right there, how might you define the growth characteristics of this microbe? What's that? It might need salt. If it needs salt, what would be the term that we give to it? Halophile. Maybe halotolerant. We can't really tell from this, but you can at least say that it's a halophile. Okay? And that's you would probably need salt in the media to get it to grow. Does this then make such a media selective or differential? And it does make it selective, because the way that I like to think of it is you're selecting for a subset of bacteria. You're selecting for the growth of a subset. Or another way to say it is you're selecting against another subset. So you're selecting for growth of halophiles and halotolerant organisms. You're selecting against the growth of non-halophiles, ones that can't grow in the presence of high concentrations of salt. Okay. We went over the first calculation here for this, and if I'm not mistaken, we said was 2.67 times 10 to the fourth, or 26,000 CFU per mil. What's the value for the second one? Anybody can get this one out. What's that? 32 million. 3, 2, 1, 2, 3, 4, 5, 6. Or 3.2 times 10 to the 7th. Anybody get anything different on that one? If so, let me know and we'll go through the math on it. Okay. Last one. 10 to the 6th, so 4 million, oh crud, 4,100,000, or 4.1 times 10 to the 6th. Given these results here, how would you classify the organism? In other words, under what temperature does it grow best? And then how do we use that to characterize it? Okay, 22 degrees Celsius. I'm going to start picking names today. No, Brendan, put your hand down. You got an exam. You got to save your energy for later. He's got genetics today. Yay. Yeah. Grace, 22 degrees Celsius. How would we define this one? I'll give you a hint. It's not thermophile. Austin, want to help? This one would be a psychotroph, most likely. Most likely, we would consider this one to be a psychotroph. Just based on what we see right now. Okay? So why go through this? In part because what we're going into in the next couple of days is talking about growth control, as you saw with the homework assignment. Today we're going to focus on the physical ways to inhibit growth control. And if I could summarize in one sentence or phrase how to control microbial growth, you do what's not optimal. In other words, to inhibit the growth of a halophile, an organism that needs salt to live, what might you do? Remove the salt. Add more water. How might you inhibit the growth of a psychotroph? Increase the temperature. Hence why we cook meats and why I will never eat raw oysters in all of my life. Okay? So The way to inhibit growth is just to do exactly the opposite, or to do something that is not optimal for the microorganism. One of the things that I want to point out here is when we say growth inhibition, we don't necessarily mean death of the microorganism. We can, but it doesn't necessarily mean death. It might just mean that the cells are still metabolically active, but they're not actively replicating. And I want to make this. I wanted to just make sure I make that distinction. Death, survival and the growth inhibition is dependent on probably two main factors: the degree of the exposure. So, for example, heating a psychotroph at 37 degrees does seem to inhibit the growth compared to 22, but probably heating them at 50 degrees or growing them at 50 degrees, that increase in temperature is going to be even more detrimental to the growth. Or, for example, a non-halophile. Growing them in 2% growth or salt may inhibit some growth, but 10% will do much more to inhibit. So the degree of that stressor can have a dramatic effect. The other one is time of exposure. The time that the organism is exposed to that particular condition can irreversibly damage the cells and prevent them from growing, if not actually kill them in the long run. We'll take a look at a couple of examples of that today. Okay. Who in here did the desiccation as the homework? Okay, good. All of you, back right or back left corner, you're right. So, desiccation folks, back there. Filtration people, front corner. Little movement exercise this morning. Yes, Erica, stop complaining. Let's see, what else? Heat. That was one of them, right? Radiation. Let's do radiation. All right. Front over here, and then what was the last one? What am I missing? Thank you. In the back. And what I'm going to have you do, I want you to, as a group, yes, these are large groups. I want you as a group to come up with a single definition or description of that particular growth control. And then I want you to, with the remaining time, come up with as many examples as you can. Okay? I'm going to give you like 4 minutes. Go. And, did you figure out the third one? Did you figure out what the relationship to mad cow disease was? Actually, yes. Oh. Yes. Prions, which we never talk about in here, but prions are essentially a mutated form of some proteins. Oftentimes they're associated with neurodegenerative effects. So the example, for any hunters out there, chronic wasting disease in the deer population is an example of a prion disease. Those can't be filtered out. That's absolutely right. Nice job. Filtration we use primarily for heat-sensitive materials. So a liquid suspension of antibiotics should be filtered instead of heat-treated or autoclaved, because that is going to lead to the denaturation or breakdown of that antibiotic. All right, last one.
Pasteurization.
Thank you.
Someone else didn't say that.
It's just like the rapid heating and rapid cooling to kill or to stunt microbial.
Yeah, I like the word stunt there, because it doesn't necessarily kill everything, right? What does it not work against? Spores it won't work against. Hyperthermophiles will it absolutely won't kill them. Thermophiles probably as well. Those are all perfect. Examples?
Milk.
That's one.
Apple juice.
Apple juice.
Cheese.
What's that? cheese, and basically any dairy product. And what was the other one? The egg beaters? Egg beaters too. Okay. The other thing to think about with each of these is we're going to start to talk about words like sterilization or disinfection, antisepsis, and those are terms that we'll bring up on Friday. But pasteurization is not sterilization. Sterilization means the death of everything or the removal of everything that we consider living. We actually consider filtration as a sterilization step because it's removing all bacteria, as long as we're using pore sizes that are small enough to prevent their flow. Desiccation, depending on what the degree of it is can be sterilization. UV radiation, same thing. Definitely we consider that to be a sterilization type technique. Okay? Any questions just based on the basic definitions here? Okay. Go back to your seats, and then what we're going to do is we're going to compare a couple of these methods with each other. How to make an autoclaved turkey for Thanksgiving, just so that everybody knows. Okay? So that is a thing. What allows for this rapid heating and cooling during pasteurization is you're using really metal tubing that is very prone to changes in heat, and it's very thin tubing as well, which allows for that quick cooling and quick heating of those of that milk product, for example. How does autoclaving and filtration differ from one another? Or how are they similar? Derek? Perfect. So filtration requires no heat, used for heat-sensitive material. What else? Emily? That's a great way to put it. Yeah, it's a physical barrier to prevent the passage of the microorganism. And I want to add to that real quick. Autoclaving will inactivate viruses. Filtration will not necessarily inactivate viruses. And viruses, depending on the pore size, can pass through these filters. If you've ever heard of a HEPA or a HEPA filter, HEPA, high efficiency PA filter, I don't remember what the last two are for. But basically they have these in hospitals, some homes have them nowadays as well. They have a very fine mesh that prevents not only the removal of bacteria, or that prevents the flow of bacteria, but also prevents the flow of viruses through them as well. And so this is one way when we're talking about purifying air in a home or something like that, that is one way by which that can be accomplished. I saw somebody put on their slides like vacuum bags. A lot of vacuum bags now are made with HEPA filters on them as well to prevent microbes from getting out of those while vacuuming. We will do, actually, some filtration in the lab in a couple of weeks from now. So we are going to, and I'll send some more information to you about this later on, because this is, it needs more explanation. But anybody used a life straw or any of those types of products before, where like you're in the forest and the way to get some clean drinking water, you can use iodine, you can use chlorine, or you can use these straws now that have like microfilters in the actual straw.
They also make like hand pump.
Yeah, they do. They make hand pump ones as well. We're going to test a couple of those in the lab, and we're going to determine how well do they actually work. Do they remove bacteria? Do they remove virus? So we'll take some time to play with those and test that out. Okay. That's just embarrassing. Sorry, Megan. All right. Let me see if there's any other one that I wanted to cover real quick. The last one that I want to bring up, because I haven't really, we've talked a little bit about UV. UV is a great example, works really well. I think one of the fears behind UV right now, or radiation, is the fact that it has radiation in the name. So for example, there have been many food scientists, food microbiologists that have proposed we should use ionizing radiation as a way to treat food items. Think of the salmonella that would be killed if we treated chicken with ionizing radiation. Then you could eat chicken raw if you wanted to, which is disgusting, but if you want to do that, it would be okay and it would be safe to do. But the fact that we bring up the word radiation in the name is, I think, personally, I think that there is a social stigma with that that prevents us from doing that type of growth control or growth inhibition. I just want to bring up this one again real quick, the removing water. Again, as water concentrations decrease, water is required for enzymatic function. It's required for every hydrolysis reaction that occurs. Okay, it's required for breaking down of molecules. It is very good. Bacteriostatic there means that it inhibits growth. Very good at that. Maybe bactericidal, killing bacteria over long periods of time. And oftentimes that decrease in water also correlates with an increase in that solute concentration. Does not mean that these particular items are free of pathogens or free of potential outbreaks. 2009, there was an outbreak in Peter Pan peanut butter of Salmonella typhimurium. Something like over 200, 300 people were sickened by it. And they went back and tested the peanut butter, and they could not find the salmonella. They were unable to actually isolate it from there. But they know it came from that source. Went back to the original plant, and the plant of Peanut King, the company that makes Peter Pan peanut butter, was chock full of salmonella. Okay? So these growth controls work really well. They're not perfect. They're not perfect. Last example I'll bring up. Anybody do home canning? Couple? Everybody knows somebody who does home canning? Yeah? Grandmas, grandpas, weird people, etc. What methods of growth control do we have in canning? Heat's a big one. And oftentimes, if you can, what's recommended is that you pressurize heat. That the pressure, so pressure cooking, will increase the temperature of that water above 100 degrees Celsius, therefore allowing for the killing of spores. And most often when cans go bad, it's because there are endospores that have survived the heat treatment. Now they're in this nutrient-rich media of whatever you've canned, tomatoes, potatoes, whatever, pickles. And because they are in that nutrient-rich media now, they're able to grow. And so if you've ever had, for example, a can burst on you, or ever been to the grocery store and seen one with like a bulge or something like that coming out of the side, that's an indication of gas production from an organism that once was an endospore. Most likely, Clostridium botulinum or Clostridium tetani, because they are soil-borne microbes. All right. One of the things to think about then is, how do we treat? So not just what are these treatments, but rather, how would we inhibit the growth? So I'm going to pull up a couple of examples here. Or I'll just read out a couple of them. 3. So how might you inhibit the growth of a mesophile on chicken? That's one. The second one is an acidotolerant, halotolerant thermophile. We'll start with just those two.
You say that last one?
Yeah, an acidotolerant, halotolerant thermophile. how might you inhibit the growth of both of those? Or kill them? I'll give you like 2 minutes. Feel free to talk it over with someone else. To inhibit chicken, or chicken on bacteria. Bacteria on chicken. Cook it. What else? Nuke it. That works too. I do not like my chicken nuked because it comes out rubbery, but you could do that and it would kill the bacteria. What else? What's that? You could freeze it, and that actually might work to, over time, kill bacteria. Absolutely. E. coli is on a surface of something. If you freeze it, the die-off is pretty quick. If it's embedded within the food item, the die-off is actually slower. Within the food matrix, it is so called. What else?
Dry it.
Fry it?
Dry it.
Dry it. Yes. Chicken jerky. You could use UV if we were talking about just on the surface. Inside, ionizing. Any of those work? Perfect. Acidotolerant, halotolerant, thermophile. Ashley? Extreme heat. Sear it. What else? Nothing else?
Well, if it's a liquid, you could filter it, right?
You could filter it, absolutely. I don't know. It's sure. This lecture, probably more than any other one, makes me hungry all the time. Some people may shun food for the next couple of days, though. Basically, do anything besides its optimal, right? So if it's halotolerant, that means at some salt concentration, it's not going to, or at some salt concentration or water level, it's still going to be okay, but if you were to super dry it, it would probably die out, for example, or something like that. I wanted to bring up something. Oh, I've got another one. But we'll cover that in a second. Here are 3 more. Different terms. Vacuum packaging, beef jerky, raisins. What's the methodology being used for each of these to inhibit growth? So how about vacuum packaging? Oh, does vacuum packaging mean desiccation? It's removing all the air. So what does that work to inhibit? Obligate aerobe. It removes the oxygen and would inhibit obligate aerobes. How about microaerophiles? If that vacuum packaging removes all of the oxygen, then yes. So we vacuum package, for example, deli meats, other frozen items, not only because it prevents them from getting freezer burn, but also because it inhibits what we consider to be spoilage bacteria. Pseudomonas fluorescens is a great example of an aerobic or an obligate aerobe that is considered a food spoiler. What about beef jerky? Ashley? Yeah, salt, dehydrate, banana chips if you would prefer a food version, sun-dried tomatoes if you would prefer another food one. How about raisins? True. Anything else? So, at least in the way that they used to be made, I don't know about these days, but raisins were dried in the sun. And so not only did you have desiccation, but you also had UV as another form of growth control there. Absolutely. Yeah. Okay. One last problem. I need to get my colors correct, though. Yeah, it works. All right. Who likes their steaks medium rare? Okay. So here's a steak, cross section of a steak. It's not the greatest of steaks, but it's a steak. Okay. There's medium rare. Why is it safe to eat a burger or to eat a steak medium rare but not a burger? From the food health perspective, why is it safe to eat a steak medium rare but not safe to eat a burger medium rare? Ashley? You're on the right track. So think about how these two are prepared. If you butcher a cow, you get your steaks, right? Steaks are just fresh cuts, no grounding or anything ahead of time. Where is the bacteria most likely to be located on a steak? On the surface. Where, if you're cooking this on the grill, what gets heated the hottest? The outside or the inside? The outside. This is why you can sear a steak and basically leave the inside raw if you wanted to. Is because the bacteria are located on the outside surface. That direct heat allows for the killing of basically all microorganisms on the surface there. Okay. Ground beef, on the other hand, is prepared differently. Ground beef, you take that steak and you put it through a grinder. And it mixes everything up. When you cook those on the grill, bacteria now aren't only on the outside surface, but they're in the middle as well. So for ground beef, oftentimes it's recommended, you need these to get up to like 165 degrees Fahrenheit on the inside. That's a temperature that if you've reached that temperature, it's enough to kill, and it will retain its heat long enough to kill any of the microorganisms on the inside as well. So keep that in mind as you're having a steak or a burger over the long weekend. Steaks, medium rare or medium well is fine. Burgers, medium well. Medium well. Okay. And anybody know tiger meat? Anybody heard that term? Not a real tiger. It's a term for like eating raw ground up beef. This is a thing. Apparently it's a thing in northern Wisconsin. Okay? Yes, people have gotten ill from this. Actually, one of the lecturers in microbiology over at the UW, him and his family got sick a number of years ago over eating round raw beef. And I don't know why they didn't have second thoughts about it, given the fact that he's a microbiologist. I guess it was just tradition. But anyway, I thought that I would bring that up. Okay, before we go. One more example. All right, so the bottom one on here. You are working with Oscar Mayer to develop methods to block the growth of a psychotrophic microorganism on cooked deli meats. The organism is also halotolerant, a nutriophile, and a facultative anaerobe. Come up with a method to inhibit its growth. We'll vacuum seal.